In a new study under consideration at Virology and posted to the Research Square * preprint server, researchers tested the hypothesis that severe acute respiratory syndrome coronavirus 2 accessory protein 7b forms oligomers with ion channel activity in lipid and detergent environments. Background: among coronaviruses, SARS-CoV genomes encode the highest number of group-specific accessory proteins with transmembrane domains, viz. 3A, 6, 7A, and 7B. One of these accessory proteins, the 7B, is 44 and 43 residue long in SARS-CoV and SAR-CoV-2, respectively, and has a fully conserved transmembrane domain, which suggests a functional role for this protein. The researchers of the present study, thus, speculated that P7B plays a vital role in the clinical manifestation of infection and SARS-CoV-2 pathogenesis. About the study. In the current study, researchers demonstrated the oligomerizing behavior of the 43 residue-long SARS-CoV-2 P7B with ion channel properties in lipid-like membranes and the presence of detergents. They obtained 7B protein as a crude peptide and synthesized, lyophilized, and purified it in-house before reconstitution in the endoplasmic reticulum Golgi intermediate compartment like lipid bilayers. The sedimentation profiles obtained using the analytical ultracentrifugation sedimentation velocity were analyzed in SEDFIT using the C model. For AUCSV experiments, the researchers reconstituted P7B samples in 5 mm myristyl sulfobetaine micelles, with or without 2 mm redundant, such as trisphosphine, and in the presence of 29.4% deuterium oxide to abolish the influence of detergent buoyancy. The research team used the same instrument and buffers for their AUC sedimentation equilibrium experiments, as were used for the AUCSV experiments. They measured absorbance at 280 nanometers after 24-hour equilibration at centrifugation speeds 23,000, 28,000, 34,500, and 42,000 rpm. The resultant sedimentation profiles were tested with several self-association models and plotted using the GUSA software. Next, they used the AlphaFold 2 server to build the preliminary dimeric model of full-length SARS-2 P7B. Additionally, this model was used to draw inferences from molecular dynamic simulation in the presence of a model lipid membrane. Subsequently, they formed 2 different tetrameric models and performed MD simulations of 100 NS for both tetramers placed inside a 1-palmitoyl, 2-oleoyl SN glycero, 3-phosphocholine lipid bilayer. Lastly, they used planar membrane electrophysiology to test the ability of full-length SARS-CoV-2 7B to form ion channels in lipid bilayers. Study findings. The infrared spectrum of P7B reconstituted in the ergic-like lipid bilayers showed a highly symmetrical and narrow amide I band centered at 1656 cm1, corresponding to an helical conformation. Also, the researchers found approximately 23 amino acids resistant to hydrogen deuterium exchange, indicating a single TMD in P7B. In the presence of reductants, they detected only monomers during sodium dodecyl sulfate gel electrophoresis of SARS-CoV-2 P7B. Contrastingly, under non-reducing conditions, they observed multiple 7B oligomers, confirming the presence of disulfide bonds in P7B. The SE experiments yielded the best fits for 7B protein in monomer-dimer tetramer, and dimer tetramer forms. For the 7B TM, in the presence of TCEP, the model monomer-dimer was the best fit, suggesting that the TMD alone could drive dimerization in the absence of disulfide bonds, but not tetramerization. Overall, this suggested that the extramembrane domains could alter the stability of higher oligomers, and the helix-helix orientation in the dimer did not require disulfide bond formation. The authors observed the P7B induced current activity at a voltage range of ±10 to ±100 MV. The addition of 7B protein diluted in 50% acetonitrile induced infrequent, unstable currents lasting several minutes and presented sequential current transitions, but did not rupture the membrane. Conclusions. The study findings showed that although the P7b had an helical conformation, it is likely that its extramembrane domain forms strands responsible for the stability of the oligomers. In the presence of a reductant, only monomers were observed in the SDS, confirming that disulfide bonds were present in the sample and that disruption of these bonds affected oligomerization, but did not prevent tetramer formation. Thus, the monomer-dimer tetramer emerged as the best fit SE model. Overall, the study provided the much-needed preview into the oligomerizing behavior of the SARS-CoV-2 accessory protein 7b, but unfortunately could not clarify its role in the viral life cycle and pathogenesis. Important notice. Research Square publishes preliminary scientific reports that are not peer-reviewed and, therefore, should not be regarded as conclusive, guide clinical practice, health-related behavior, or treated as established information.